Hello everyone, hope you all are doing well. So today we are going to discuss the next question of our agriculture optional answer writing practice series, and the question for today is, what do you understand by somatic and meiotic crossing over, and discuss the mechanism of meiotic crossing over. So we will see both of the things. So let's see how we can approach the answer. So, and crossing over is an important topic. So crossing over and segregation, so all are these are very important topics. So please do them very well. Prepare them very well. So in introduction part of this question, you can simply mention or you can simply define what crossing over is. Simply mention about crossing over. So crossing over is the process of exchange of genetic material. Process of exchange of genetic material between two homologous chromosome, or you can simply mention between homologous chromosome, between homologous chromosomes, and it is a key event that promotes genetic variation. It promotes genetic variation because there is some exchanges between the chromosomes, so obviously it will create some variation. Now first of all we will see about what somatic crossing over is. Somatic crossing over, so it refers to the exchange of genetic material between two homologous chromosome in somatic cells. Okay, so it is crossing over in, simply you can mention crossing over, that is exchange of genetic materials between two homologous chromosomes. We have just defined what crossing over is. So it is crossing over in somatic cells. Crossing over in somatic, or you can mention somatic cells as also as unreproductive cells, or you can say non-reproductive cells. Crossing over in somatic or non-reproductive cells. But instead of writing crossing over, you simply write that the definition of crossing over, it is the process of exchange of genetic material between homologous chromosomes in somatic cells. In bracket you can also mention that these cells are non-reproductive cells. And if you see the occurrence of somatic crossing over, it is very rare and usually seen in very certain organisms. For example, it can be seen in Drosophila. It results to meiotic recombinations. Okay, it can also lead to genetic abnormalities. You can also mention here that it can result to meiotic recombination recombination, and it can also lead to genetic abnormalities. In genetic abnormalities, you can mention examples how it can lead to genetic abnormality is the lack of heterozygosity, or you can say loss of heterozygosity, and you can mention examples such as of cancer can be due to this somatic crossing over. And if you want to write its mechanism, you can also mention that it occurs during mitosis phase. Occurs during mitosis phase, typically in G2 phase of cell cycle. Okay, and the mechanism is similar to meiotic crossing over but not lead to gamete formation. Okay, so this somatic crossing over, you can also mention it is also known as mitotic crossing over, and it is very similar to kind of meiotic crossing over, but it does not lead to gamete formation. It does not lead to gamete formation. So these are the points which you can mention in somatic crossing over. I hope you understand this. Now, what is about meiotic crossing over? If you have a general knowledge of mitosis and meiosis, we all know that gamete is formed in reductional division, that is meiosis. So obviously in mitotic crossing over, there will be no gamete formation. Now the second type of crossing over is the meiotic crossing over. meiotic crossing over. So it is the kind of, you can mention, the reciprocal exchange of genetic material between, it is reciprocal exchange of genetic material between two non-sister chromatids, or you can simply write between non-sister chromatids of homologous chromosome. You can also mention the time, that is in or during prophase 1 of meiosis. So it is kind of also crossing over only, but here the exchange of genetic material between non-sister chromatids of there is exchange of genetic material between non-sister chromatids of homologous chromosome during prophase 1. So you can mention these things. You can mention the functions of this meiotic, not mitotic, sorry, mitotic, so meiotic chromosome crossing over. So the functions of this meiotic crossing over is that it creates genetic diversity among gametes, increases genetic diversity among gametes. Secondly, it as ensures proper alignment and segregation of homologous chromosome, ensures proper alignment and segregation of homologous chromosomes. So these are the major functions. So this was about somatic, that is mitotic chromosome and meiotic crossing over. Okay, somatic crossing over and meiotic crossing over. Now in the second part of the question they are asking us the mechanism of meiotic crossing over. So we will discuss that mechanism also. Mechanism of meiotic crossing over. So the actually the first it generally, you should remember first that it occurs in prophase first, and particularly in Pachydine stage, but we will see the in detail. So first, in the first process there is pairing of homologous chromosomes, pairing of homologous chromosomes. So this generally happens, this process starts from diplotin stage and proceeds till zygote stage, from diplotin to zygote. Here homologous chromosomes start aligning. Synapsis occurs with the help of synaptometal complex. So all these things you can mention, but simply mention that it is here the pairing of homologous chromosomes happens. Second process is the formation of synaptonemal complex. Formation of synaptonemal complex. So this starts from zygote and proceeds towards pachytine. So zygote to pachytine. So here obviously the synaptonemal complex which forms holds the chromosomes tightly and tightly together. The chromosomes are not now called as bivalents or tetrads. So it is the second process. Now third is the initiation of crossing over. That is the crossing over starts. Initiation of crossing over, it happens in Pachydine stage. Okay, so here you will see that the double stranded DNA or some anything double stranded things or chromosomes, you can simply mention, double stranded breaks are introduced by enzyme SPO11. So here there are double stranded breaks. Okay, wherever there are double strand, so they create kind of breaks on those double strand using the enzymes SPO11. Okay, then one strand inverts the homologous non-chromatid forming a D loop. So suppose this is two chromatids, so it one inverts the non-homologous chromatid and sorry, not non-homologous, instead homologous chromatid and forms the D loop. This all things we are talking about the third stage. So after the formation of D loop, there is exchange of strands and DNA synthesis occurs. So kind of you can remember these things. Now the fourth stage is that formation of holiday junction. Formation of holiday junctions. So cross strand pairing leads to the formation of holiday junctions, which are cross shaped structures. So what happens during the chromatin, the exchange of chromosomes in those chromatins, a cross-shaped structure is formed, which is known as holiday junction. Then there is branch migration and dissolution in the diplotin phase. Branch migration and resolution. This happens in diplotin phase. So here the holiday junction migrates, increasing the region of hybrid DNA. The junction is resolved by endonucleases, because we all know that endonucleases and will cut junctions from several ends and it will resolve the junction. Okay, there could be other horizontal junctions. Okay, non-crossover products will take the horizontal shape and the crossover products will take the vertical shape. So this horizontal shape, vertical shape is for crossover products and horizontal shape for non-crossover products. So this will happen that all the year you can see branches are there. So this branch will migrate and they will form certain resolutions. Okay. So this is the fifth stage. Now the sixth stage is chiasmata formation. Chiasmata formation. So the physical evidence of crossing over is visible as chiasmata. So these help hold homologous together until anaphase 1. So these are the things which we can mention, and in the conclusion part you can mention about both the crossing overs and how they are involved in genetically significant process ensuring variation and proper chromosome segregation. So these things will be more than enough. Although these topics will you will feel these topics are a bit complex, but yes, we all know that things like that these are bit complex in nature, but understanding what mitotic crossing over and meiotic crossing over is will help you, because these types of questions are expected, or either you can leave these types of questions because this will be generally asked in options. So you can leave, or you can simply try to remember these processes, remember some basic facts by which you can answer the question. So that is all for today. If you like it, please press the like button, please do share, and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis. And if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.